Well, I'm very glad to see so many young people here, and also quite a number of elderly gentlemen as well. Because I think this topic of evolution is in fact one of the great intellectual challenges that science has to meet. Because of what it is, we are engaged in basis of investigation which has to reconstruct the past. Of course, very little of the past is with us. But fortunately, there are signs and remnants that have been preserved. And so this is what the aim of this is. Now, of course, if you were to tell someone you could just come from just ordinary matter and create the president of NTU out of this, they wouldn't believe you. But as you will see, it has been done without anybody running it. That is, the machine has no pilot. There has been no supervisor. It has just come that way. Many years ago, the logician and philosopher Karl Popper declared that evolution couldn't be a science. And he claimed this because he had one principle which most people found perverse. He says the only thing that creates a science is that you can disprove it, any theory, by experiment. So that means if you have a theory which you've tried to establish by experiment, the last thing you're going to do is to do experiments to try and disprove it. But that is why he said evolution can't be a science, can't be an experimental science, because we can't run it again. You can't go back to the drawing board and do it again. If you make mistakes, if mistakes have been made, they are with us forever. Now, this idea, I think, is very important because the tendency that people have, because of their intelligent brains, is that there's always got to be a designer. Someone must have worked it out before, because otherwise it wouldn't work. It won't come about. But as you will hear over the next 10 months, you will hear how it has all taken a long time, 10 to the 10 billion years, and it has proceeded stepwise through this. Now, of course, there is something that many people wanted to know about evolution. Was it always driving onwards and upwards? Was there always going to be improvement? Was there always going to be some new thing happen? And in fact, that was a very famous social philosopher, Herbert Spencer, who believed that everything was being improved by this. And that was the principle. Evolution was always to get better, to be more honest, to be everything that was desirable. But as you will discover, as you listen to these talks, at least for the large parts of evolution which are biological, there is no purpose in it. It is not purposeful. There was no creator who created man, as one of the stories goes, and puts him in there and did it all. So it is believed in a document in one day. He created all the animals in another day. And so that is how most people, most minds thought about it. The big lesson to learn in science is that only one subject is the art of the perfect. That's mathematics. Physics is only the art of the optimum. But biology is the art of the satisfactory. If it works, you keep it. If it doesn't work, that's it, it's gone. That is, it's not an improvement. It is only that necessary step to be taken to keep it going. And I think that is very important, that we drive out all thoughts that we are aiming at perfect animals, perfect plants. It's not. They just are satisfactory. They work. Some of them even don't work very efficiently, but they work. They work enough to survive. Now, as you know, the great prophet of at least biological evolution was Charles Darwin. He was the one who saw and described what he called natural selection. That is, those that were fitter for reproduction and survival survived. There was another later on, a man called Kimura, then discovered what he called neutral selection. Now, I tell you what that is. That is the survival of the survivors. And in fact, in the early 1900s, you could play two games with balls colored in the following way. This is part of what Ehrenfest put together. So let's imagine we have a big bucket and we have balls labeled from 1 to 100. And we play this game. We remove a ball at random, and then one of the other balls at random is selected to duplicate, so that the total number of balls remains at 100. Now, what you will find is if you play this game for a long time, eventually all the balls in the urn will have the same number. Because naturally, when you've picked one at random, then that number has twice the probability over all the others of being selected. And if it does, then it will just climb. And so that is the survival of the survivors. But let us suppose that one of these balls has just a better chance of duplicating, is a better duplicator than all of the others. Again, once we withdraw something, we will copy that one. And you will now see that that one will dominate the whole thing because it climbs as follows. Now, what's the difference? If we are observers and didn't know anything, that is the difference between the survival of the survivors and the survival of the fittest. And how do we decide? We have to play the game several times. In the first case, we'll end up with a different number each time. It'll just be by chance. In the second case, the fittest will survive. So, of course, you must be careful that when you are looking at the evolution of various animals or plants or bacteria, that you are not dealing with cases where you just happen to have survival of the survivors. So it's very important to understand what is fitness for survival and reproduction. That is the one thing. Now, it so happens that the most remarkable access to the materials of evolution have come about very recently, last 20 years. And this is by our ability to sequence, to find the DNA message written on the whole genome of lots of different bacteria, lots of different animals, lots of different plants. When I started to work on this, very little was known about this, and I began to collect these sequences and began to study them. But I decided, for another reason, that most of your genome is junk. Now, I got to give you that definition. About 1%, 1 or perhaps 2% at the most, of your genome is involved in directing the amino acids to form proteins. Since the behavior of all your cells is by the proteins that constitute your cells, whether it's brain or liver or whatever it is, then it's very important that that is the one that turns out to be the real driver of evolution. Now, many people didn't like the idea, but I think you will find out, if you study many languages, that there are two words in many languages that separate, that make the distinction between junk and garbage. There are two kinds of rubbish. What is the one kind of rubbish is the rubbish you keep, is called junk. The other kind is the rubbish you throw away, it's called garbage. In Japanese, it's gomi and garakuta. Garakuta is a junk shop. All right, now, I discovered this in my own actions, and it's very important. And I want to tell you this in the form of a story. When we first started to work and we got a Spinco centrifuge, ultra-centrifuge, came in a beautiful box to the lab. And so I took the box home, because it came from California in this box. I took the box home, and I said to my wife, I'm going to keep this, and I'm going to make a bookcase out of it. And of course, it was put in the attic of our house, where we kept it. And after a while, about 12 years, I came home and my wife said, "Oh, by the way, I threw your box out, your planks out." I said, "Oh no, I'm going to make a bookcase." She said, "I know you never will." You see, and so I threw it out. Go and buy yourself a bookcase. So I had kept that as junk. Just happened to be, no, but it wasn't doing any harm. And I was going to use it. But my wife, who was one of the strong agents of natural selection, decided to convert it to garbage and threw it out. So I think you need to have this distinction. Now, of course, being me, I had a purpose for this junk, but my wife knew me very well. She knew I'd never get around to doing it. I would always have something else to do. So it was just sitting there. Wasn't doing any harm until she decided she needed the space. Well, you have to accept that your genome is 98% junk. It's just something you've carried around with you. It's not going to be of any significance. And this people do not like, because they think that everything has got to be there for a purpose. That is, it's put there. If it's not functioning, it'll get lost. Yeah, this junk does turns over, and sometimes it gets deleted. Sometimes there's more junk comes in from other sources. So, why is it that we carry this load of useless apparatus? If you take certain fish, like the famous fugu, they have a genome, although they are higher organisms, much like ourselves. They have about the same number of genes as we do, but their DNA is 8 times smaller than ours. So they just happen not to have as much junk. And for a long time, we tried to persuade people, when sequencing was very expensive, that we should do the fugu genome. And fortunately, we were able to do it here in Singapore in 2002. It was one of the first big genomes that became available. I give you the story because for each of these stages, once people are confronted with the information, they think it's paradoxical, and it can't be that way. So when we read genomes, when we notice that when you make proteins, certain of the codons, this is something that we've known for a long time, certain of the codons, the third position doesn't matter what base it is. It can be either a mutant or not, as the case may be. The mutation is called a transition, so it can be C or T. And this turns out to be very powerful, because that doesn't matter. You can make exactly the same organism whether you have a mutation or not there. And that turns out to be an internal clock in the genomes of all organisms. because this goes right from bacteria all the way up to you. So by looking at this position in those codons, you can give a description of the whole of evolution, starting from bacteria right up to man. And it's very remarkable what you can discover from that. You first of all discover very quickly that lots and lots of organisms, not ours, but lots of others, lots of bacteria, are at equilibrium. They're not going to change anymore. That's just the way they've done this, they've done their evolution. So that begins to make the argument that you could stay the same way and just, mark the passage of time by this process of mutation and not change at all. And as you know, there are at least 2 animals known which have survived for nearly half a billion years, and who have the same structure as the fossils which were dug out. One is, they both are fish. One is the elephant shark, which again is something that we've worked on extensively in Singapore. This is right at the bottom, and it's just survived. The skeleton is exactly the same as the fossils retained. And the other is the famous coelacanth, the fish at 300, last found 300 million years ago. Now what I think, why don't we look to see if we can extend this? So what about these bacteria that live in incredible places? For example, just give you one of them. It lives in water, and in fact it lives in the water that comes out of the vents at the floor of the ocean, which are full of hydrogen sulfide, and the water emerges at 135 degrees centigrade. That is, it's boiling water. And these bacteria will grow in the lab in boiling water. They grow without oxygen. They can do all sorts of things. So, they've probably been around there since the dawn of life. And there are 2 kinds of these bacteria. And they are very probably exactly the same as they were, I would say, 1 billion years ago when life began. So, it's probably 2 billion years ago. So what I think we have to be careful of is that everything is not moving onwards and upwards. Everything we can dredge out of environmental places. We can find organisms living there, and they probably haven't changed their existence for all this time. So you could ask yourself, what is the minimum number of genes you need in order to be a total independent, make everything from the environment? And it turns out that we can estimate that at about 1,800 genes. So call it 2,000. So there's 2,000, you can synthesize DNA, you can repair DNA, you can recombine DNA, you can make proteins, you can do all kinds of catalysis, you can preserve your membranes. 200 is the basic thing. So we now take another common bacterium, E. coli, another one that we all have worked with. And we count up it's got 4,000 genes. So that's another 2,000. What is E. coli doing with these extra genes? Now, the clue to this is to just think about what E. coli likes to do. E. coli likes to live inside your intestine, where it gets a lot of food for free. Doesn't have to make it. So that means that anything that it has made, that it gets for free, it can switch off. And so what E. coli has done is to construct a whole regulatory machinery of genes that enable the genes to be turned off. And this is incredible because it's very expensive to make the things in bacteria, and it's very expensive to make the plant, the factory that you need to make things. So if you can get it all for free, you can actually then have an improved growth. And it has then accumulated, and this is the source which everybody wanted to know. If you have gene regulation, why wasn't repression? That was what they discovered. The first regulators in bacteria were ones that switched off genes. It turns out that that's exactly what you want if you want to enjoy the privileges of being a parasite, but still retain the ability to be outside, independent. Of course, many parasites have come about and then have lost their genes, so they live in you forever. So you must distinguish between the parasites that have now lost everything because they didn't need it. That's just natural selection. Wouldn't affect their survival if they lost the ability to make things. And so we have these. They call mycoplasma. They're not very primitive. They are degenerate. And that's what you have to make a big distinction between, because parasitism, which is now grows throughout the whole of evolution, because everybody can get a free ride, they'll do it. They'll get it. Malaria, we have a whole lot of organisms that live inside animals or live inside plants because and they've adapted to this. So once you look at evolution in this way, the big thing to remember is its mutation that'll drive it, and the ability to get DNA for free by taking in other organisms' DNA. Now, I'll tell you a remarkable thing. One of these animals, one of these bacteria that lives in boiling water has got machinery in its membrane to take DNA from the water. So, if you like, sex is through single molecules of DNA. And just think, boiling water means enzymes are not going to survive very easily. And they have then taken this in and can incorporate it into their own DNA. So by assimilating DNA through various entities, like DNA directly, or through viruses, or through anything else, you can expand the genome. You can then find the material to make new genes, because they will just automatically be selected. So I think that when you come to hear the rest of the things, the rest of the talks, you will see that it's all by chance, but the gambling is pretty good. You get a good chance to do things because you get a good chance to make yourself better. And because you are so buffered against evolution in your genetics, you are buffered there. We now have a clock inside the genome which can simply measure the passage of time. So I think that for the future, biological evolution, we will be able to reconstruct quite a lot of the previous existence of bacteria. And in every case, there have been wonderful cases of finding some remnant which shows that that's correct, that animal did exist then. So I think this is what you will hear through this. Of course, the most interesting stages of evolution is when we give up doing it by biology. And for us, man, that is something that we are doing through our daily experiences. But here it is not done automatically, but it is done because there are people involved who have intentions, who have desirable things. Fitness can often be measured in lots of different ways because people fight wars in order to survive or not to survive. But it all is in the continuity. And the questions that we need to ask ourselves as we come to the last million years of this period, from the starting of men, hominids, to their evolution into this, throughout the whole of the population of the world, through the invention of humans, of tools, and of science itself. I wish you a very pleasant 10 years, but keep your brains active. And I should just add that I did this once before in Lisbon with the Gulbenkian Foundation. But we didn't have 10 months. We did it all in 2 days. And some of the people that spoke there are back again with us in this period, having done much more than they did. This was about 12 years ago. But it was, again, to see the entire of history over 10 billion years. So there's your enjoyment for the next talk, and the talks that follow. Thank you very much.